This video will discuss on DNA transcription. At the end of this video, students should be able to describe the process of DNA transcription. First, let us define the meaning of DNA transcription. In the dictionary, to transcribe means to represent speech in symbols, or to write out speech in another language or alphabet. DNA transcription is defined as a process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. Among various RNA types present in cells, only mRNA will get translated into protein. Now let's look at the process of DNA transcription in prokaryotes. We choose prokaryote as a model because the process is much simpler but comparable to what happens in eukaryotes. DNA transcription involves three phases of initiation, elongation, and termination. During initiation phase, the DNA molecule will be prepped prior to the actual transcription process. This involves recognition of the promoter region and the transcription start site, assembly of the transcriptional machineries, and unzipping of the DNA double helix. The important enzyme for DNA transcription is RNA polymerase. Its core enzyme composed of 5 protein subunits, namely 2-alpha, 1-beta, 1-beta prime, and 1-omega. This core enzyme cannot initiate transcription at the promoter site. Rather, it can only elongate or extend the transcription chain. Recognition of the promoter region requires a sigma factor. Together, core enzyme and sigma factor form a holenzyme. The promoter region in prokaryote chromosome consists of pribnobox at -10, and a CG-rich region at -35, upstream to the transcription start site. Promoter region controls the rate of gene transcription. Promoter activation increases transcription, while promoter inactivation reduces transcription. RNA transcript will be produced according to the template strand. The sequence of RNA transcript will be complementary to the template strand, and exactly similar to the sequence in the coding strand. During the initiation phase of DNA transcription, RNA polymerase holenzyme will quickly scan through the DNA for the gene of interest. Sigma factor will help to identify the promoter region of the gene of interest. Then, the holenzyme will bind to the -35 sequence to form a closed initiation complex. The holenzyme will then move from -35 to the pribnobox. The AT-rich sequence in pribnobox will melt and produce a transcription bubble. Both holenzyme and the transcription bubble form an open initiation complex. The holenzyme will continue to unwind the DNA double helix downstream to the gene transcription start site. As the enzyme opens up the DNA, topoisomerase will relieve the supercoils. A short DNA-RNA hybrid helix consisting of approximately 10 bases will be synthesized by the holenzyme. Synthesis of this short sequence does not require a prime or primase. The hybrid helix will be separated shortly after synthesis. The elongation phase will take place right after the short DNA-RNA hybrid helix synthesis. In this phase, ribonucleosides will be added onto the short RNA strand, one base at a time. This sequential process will elongate the initial short RNA sequence to form a strand of RNA transcript. As the holenzyme leaves promoter region, it moves downstream along the template strand and adds more ribonucleosides to elongate the initial sequence. Addition of ribonucleosides requires energy from the phosphate group and follow the Watson-Crick base pairing rule. The elongation process of RNA transcript occurs in a 5' to 3' directions. Unlike DNA polymerase III of the replication process, RNA polymerase holenzyme does not possess proofreading activity. If a wrong nucleotide is added, the holenzyme will backtrack, cleave the transcript, and restart the whole process again. Therefore, DNA transcription has a higher error rate than DNA replication. After the whole gene has been transcribed into RNA transcript, termination phase will occur. The ending of transcription process will take place when the termination signal is reached. The termination signal has a unique sequence. When transcribed into RNA transcript, the sequence is self-complementary. This causes the RNA transcript to fold onto itself and forms a hairpin structure. The termination signal will be recognized by RNA polymerase holenzyme itself. However, for some genes, a rho factor may be required to correctly recognize the termination signal. Now let's have a look at some of the post-transcriptional modifications of RNA transcript. Prokaryotic messenger RNA requires very little to no modification. These mRNA will be translated by ribosome right away. This is because prokaryote lacks nuclear membrane. There is no separation of DNA molecule from ribosomes. Meanwhile, transfer RNA and ribosomal RNA will be transcribed in long unit. These RNA transcripts will require further processing and modifications to form mature, functional tRNA and rRNA molecules. As for eukaryotic mRNA, several post-transcriptional modifications will occur. These include removal of introns and splicing of exons. Introns are non-protein coding sequences in between protein coding sequences of exons. Eukaryotic mRNA will also be stabilized by addition of 7-methylguanosine cap at the 5' end and polyadenylation, or poly-A tail at the 3' end. These modifications transform initial mRNA transcript into a mature mRNA. 7-methylguanosine cap and poly-A tail protects the mature mRNA against the attacks by 5' to 3' exonuclease, and 3' to 5' exonuclease respectively in the cytoplasm. These prevent mRNA from being degraded. Additionally, they also facilitate the translation process. This marks the end of this video. The next learning activity will be conducted through a team-based learning or TBL. First, we will conduct an individual readiness assurance test or IRAT. Students are required to answer the following questions without referring to the notes, lecture videos, or textbooks. Then, students are required to form a group with peers, with at least 2 persons per group. Please discuss within your group and answer the same questions again, without referring to the notes, lecture videos, or textbooks. Thank you for your participation.